Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor for today, Quanterix Corp., I'd like to welcome you to Personalized Medicine, the Need for Precise Protein Quantification and Identification. My name is Elizabeth Lamb, and I'll be the host for today's event. At this time, I'd like to introduce our moderator for today's event. He is Kevin Hrusovsky, the CEO of Quanterix. Welcome, Kevin.
Thank you very much. And we'd like to welcome everyone to this webinar. We're pretty excited about it. Just in way of background, we are a company that is working to digitize biomarker analysis to advance science of precision health. We also lead a Precision Health Summit each year. On October 24th and 25th, we expect to have about 40 presentations ranging from oncology to neurology to inflammation and cardiology and even infectious disease. That's going to be October 24th and 25th in the Boston area. So please look us up, and we'd really like to have you attend. We actually have about 80 assays now off the shelf, and we've done about 280 total, meaning the rest are homebrew, and we have an accelerator lab where we've actually run 325 studies to date. It's a very rapidly growing part of our overall franchise. And today we're really pleased that we're hosting this webinar with Dr. Jennifer Van Eyck. Dr. Van Eyck is Professor of Medicine at Cedars-Sinai Medical Center, Director of the Basic Science Research in the Barbra Streisand's Women's Heart Center, and Director of the New Advanced Clinical BioSystems Institute, where she recently moved from Johns Hopkins University. Most recently, she has become the co-director of the Cedars-Sinai Precision Health, focused on in-hospital and population individualization of healthcare. Dr. Van Eyck is internationally known and a leader in the area of clinical proteomics. Her laboratory is well known for the extreme technical quality of data generated, rigorous quality control with tight percent CVs while applying these to key clinical questions. The aim is to maximize throughput and reproducibility in order to move targeted and robust discovery methods into large population, healthy continuous assessment and clinical grade assays focusing on brain and cardiovascular diseases. Today, Dr. Van Eyck's title is Personalized Medicine, the Need for Precise Protein Quantitation and Identification. Jenny, thank you and welcome.
Thank you very much. It's my delight to be here, and welcome again to everyone who is listening. Thank you for joining us for this hour. So today I thought we'd talk about really the need for being able to be sensitive and precise in the context to precision medicine. So precision medicine really has two sides, and that one is to have precise therapy, meaning we're going to get drugs on targets, or at least therapy is on target, doesn't have to be a drug. And the other side is really being able to diagnose or risk stratify or prognose an individual to understand what their disease status is and how they'll respond to any therapy or intervention as required. So one of the premises in our lab, because we are very protein focused, is that the proteome of a person, all that diversity within a cell and across all the different cell types that make up our bodies, really will reflect both the past, so your history, your family history, what you've been exposed to, as well as who you are right now, and that will dictate how you respond in the future, be it to other environmental responses or to therapy or to other diseases as you obtain them as you grow older. And that if we can understand this interconnection, this complexity of biology, this signature that is you, we're going to be able to better understand the disease process. And right now we're not at that stage, but I think that's the hope for many of us who work in this area. So one of the premise of all of this that's going to be able to enable precision medicine is the ability to accurately and robustly find assays that are able to be selective and specific, and we'll kind of go over that a little bit today. A lot of it will be focused on sensitivity and the need for sensitivity, but we'll also discuss specificity and selectivity of our biomarkers, trying again to get it so we can map individualized person's response. So On the next slide, as I said, we're protein-centric. We are starting to move into metabolomics and other areas, but we are very protein-centric. And one of the reasons we are is that we believe that the phenotyping of an individual, the molecular phenotyping, can be represented well by the proteome because it's a reflection of all of the other omics within its context if we're able to capture that diversity. And biology is really complicated because every protein can have many post-translation modifications or co-modifications, and each gene can have multiple splice variants, and there's different types of splice variants, both traditional ones as well as non-traditional ones, alternative splice variants that we are now finding out about it. And if you take all of that diversity, now we have really the ability, if we were able to capture it, a sufficient enough of diversity, biological and pathological diversity, that we should be able to capture that individualization of your particular proteome or your particular disease status, depending on what level you go to. So as I said, proteins are quite remarkable. They have many different isoforms, and for example, we'll talk a lot about cardiac troponin I. It's an isoform of the skeletal troponin I, both skeletal fast and skeletal slow. They all reside on different genes, but yet they're still considered isoforms because they're so highly conserved and also have the conserved function. But yet you can also have in many other genes actual splice variants where in the single gene you have different splice variants. And each one of those could be regulated due to disease or due to maturation. And on top of that, as I mentioned, we have post-translation modifications. And of course, those can compete for the same residue. So you can have two modifications competing for the same residue. So for example, phosphorylation of serine can also happen with O-glycoalkylation, for example. Lysine, of course, has many post-translation modifications that compete for a single lysine. And arginine also has methylation and a modification most people don't know about, citrullination, which I'll talk about later. We can have they say, I don't even know how many post-translation modifications, but they will be hundreds. And we don't even have tools to even begin to monitor really the top 20 of them. And even within those, there is such a complication, and but we do know that some have biological importance, and that biological importance will provide you with a selectivity that we don't get any other way. And of course, we have SNPs, and we know that there's many, and we don't yet understand and have not yet identified all of them. So if you're going to be able to capture this molecular complexity that is you versus myself, how are we going to do that? And I can tell you this will not be trivial, and it will probably take us multiple generations of scientists to be able to get there. But what we do know is that we assume that the proteome is very extraordinarily large. And it's also very biologically important and will reflect the pathophysiological status. But we don't yet know that the extent of protein variability, the differences between individuals, or the interplay between diseases, such as if you have diabetes versus cardiovascular, or the combination, how does that work on top of your family history? How does that change your proteome, and how can we capture that in order to understand your prognosis and also how you respond to therapy, or even if you need therapy? Precision medicine, we assume that we want to collect and capture this molecular complexity at the individual level. And, the assumptions we've made is that the specific modified proteins will provide this, and also that we'll be able to combine multiple omics potentially, or pathways, in order to be able to represent this. So it comes down, therefore, to assays, and that's about sensitivity of your assay, the specificity of your analyte or analytes, and selectivity, whether or not you need one analyte or multiple analytes to be able to go after the clinical domain that you're after. Whether or not that's an acute, we're going to talk a lot about cardiac TNI. It's a single marker, although it can be used in combination, but it's often used as a single marker and is very good. But yet when we talk about chronic diseases, it's not so clear that a single marker might be sufficient, and it's certainly not clear when we talk about the interplay between diseases whether or not a single marker will be able to do that. Most likely not. And then we have to talk about panels and the specificity of each part of that panel. Having more bad proteins or more bad markers does not make a good panel. Really having that selectivity of what you're trying to monitor and put that together as a panel, and understanding how those interplay with each other will be actually what's needed. So we are going to talk about heart disease a lot today, and in particular, heart attacks and myocardial infarctions, and also in heart failure. So 33% of the people who survive an MI will go on to develop heart failure approximately. And it's quite staggering that in just over every minute, someone will be diagnosed with having heart failure in the U.S. Equally, one person or just over one person in Europe will also be diagnosed with having an MI or having a heart failure. And there is, people argue a little bit about what number of people, what your survival rate is after heart failure, but it's not good. It's 40 to 50% within the first few years. And this has made it so that there are so many people living with heart failure, and we do not have a lot of treatments for them, and certainly we cannot cure it. So in the field of cardiovascular disease, or over the lifetime, you have a lifetime of risk. Often it's talked about the Framingham risk scores. These are risk scores that are really to do with things like lifestyle, smoking, diabetes, family history, which they can help give a risk score for whether or not you will develop CVD. But yet, along the process, there are many places that you could have biomarkers that were circulating that could help you understand better the progression of a person or an individual person in their cardiovascular disease, and some of those are listed below there. Equally, there are imaging modalities that people use to go along the top trying to figure out where you are in the spectrum of your CVD risk and whether or not there's a way that we need to intervene, if it's even possible yet. Combinations of imaging and biomarkers are often sought after, and I think this is a growing area and one that we need to be able to deal with. Hitting the window of opportunity, having the right diagnostic marker or markers in the time in which you're going to make that diagnosis is very important. Sometimes you're going to be having to talk about having population screening. Other times it's when someone comes into emergency, or being able to monitor someone from home using remote devices, for example. Here is just showing you one, of course, highlighting cardiac troponin I and cardiac troponin T. This is if you're coming into emergency, you're having a heart attack, your cardiac myocytes or your cells are injured or they're dying, and they're releasing the cardiac proteins into the bloodstream. This is a concept that's been around for a long time for this set of markers, but it is not the only markers that come out of your heart. Of course not. As you release all the proteins and you damage your cells, there's going to be a selection of which proteins are released first, second, third, how long they're released, and what their clearance kinetics are going to be. That combination is really what makes a good marker. You need to have the right protein and the right form of the protein, or the right proteins being monitored at that clinical window where a decision can be made. So cardiac troponin I and troponin T are both cardiac-specific proteins, and that's why they beat out essentially proteins like myoglobin and CKMB and FAB9, and actually tropomyosin, which was one of the first proteins ever measured in the blood as a concept of having an acute injury marker that was coming from cell-specific proteins. So actually it was tropomyosin, and then through the years, cardiac troponin I and troponin T came on board because of the release and clearance kinetics, because of the specificity of the proteins and of the antibody pairs that could be produced for them. So why should cardiac TNI be such a good marker? Well, it is cardiac specific, as I mentioned. It's also a dominant protein in the myocyte. It is one of the proteins that comprise the myofilament or contractile proteins, the subdomain. It's in very high concentration, just like cardiac troponin T is, or myosin, or tropomyosin. All these proteins dominate the myocyte, and therefore when they're being released, they're also going to be at a higher concentration in the blood than a nuclear transcription factor that also might be cardiac specific. Higher concentrations, even though these are very low concentration analytes, are still better than super, super low concentration analytes. They were originally believed to only be released upon dead myocytes, because we did not believe at the time that these markers were coming along that cardiac muscle could regenerate itself. We now know that that's not true. Heart can regenerate itself. It's just very poor at doing it, and it gets worse with age. The other important part of it, we often talk about wanting to have a biomarker that's closely related to the function or the pathophysiology of a disease, and cardiac troponin I is certainly such a protein. It is a key contractile protein. It really is the molecular switch that causes muscle to contract or relax. T and troponin T is also involved in that, but it's not the direct switch. And it can, if you actually remove cardiac troponin I from a cardiac myocyte, it will no longer contract. And if you put back a small peptide or the protein, you can reestablish contraction. So obviously it's a key molecule in contraction. So is it cardiac specific? Yes, there's still a little bit of debate, but we believe it's pretty true. Is it dominant? It certainly is, but not the only protein. And now we know that there is cardiac myocyte turnover. So now we have this concept of leaky myocardium or myocardial injury, where you can actually have continuous turnover of cardiac TNI present in the blood, potentially even in healthy individuals. And I'll show data later that actually shows that that is correct. The cardiac TNI is actually in the blood or present in many, many healthy individuals. I would like to say all, but we don't have that, of course, data for everybody in the world. But certainly for the ones that we've studied, we can detect them and quantify it using the Quanterix ELISA platform. And then we can see a turnover. So now we can conceptually think about cardiac muscle a little bit differently and what that means with respect to both having a heart attack, but also during the recovery of a heart attack and maybe risk stratification of those which will go into heart failure. So there's a number of challenges to quantify cardiac troponin I. And in the IFCC task force in 2012, this was a great task force. I highly recommend you to read it. It was said that we should be able to, if we could have everything perfect, measure cardiac troponin I or troponin T in 50% of subjects, but we should want to be able to do it preferably in everybody. Alan Rue and Rob Christensen, with their classification, actually suggested as a subsequent that we should be able to do it in 100. But this has not been feasible until recently with the development of the Quanterix ELISA platform and mounting onto that platform, which has the sensitivity, the cardiac troponin I assay. So that's what we're hoping to be able to do. And then the question becomes, if that's possible, can we exploit this additional sensitivity and help actually to define better what's going on in individuals as opposed to making the clinical domain more mucky? We don't want to do that. So this is taken from another wonderful paper from Fred Apple that shows really the percent measurable along the x-axis of people with MI versus those different assays that are out there versus the percentile micrograms of nanograms per mil. And you can see that the Abbott high sensitivity one is the closest to clinical use. Single X is getting up there. But the question is, can we get even more sensitive and pick that up in healthy individuals? That's what we want to be able to do, and whether or not that would add value. So why is sensitivity important? Well, just to remind everyone that in any analytics, doesn't matter whether or not we're talking about an ELISA, sensitivity, there is a lower limit of detection, a lower limit of quantification, and an upper limit of detection and an upper limit of quantification. In between that, there should be a linear range, and that's where you want to be measuring your analytes, if at all possible. That's where you get the biggest full change or biggest change in the readout, in the measurement, for change in a concentration. So you're going to be more accurate in this linear range. What happens, however, if you have analytes, let's say the black X's, you're going to be able to, in the middle of the range, you should be able to get good quantitative data on every single one of those individuals. However, if that's at the bottom of the curve, close to the LLOQ, then you're going to have missing data, and you're going to have data that you are below quantitation or below the limit of detection, and you just don't know what those values are. You don't know if they're close to or far away. So missing data, this often happens when you're measuring cytokines or TNI at low levels. And before we had super sensitive assays, we thought healthy people didn't have TNI in their blood or cardiac TNI because the detection limit did not allow us to see any. It was below the LLOD. So what you want to be able to do instead is you want to be able to increase your sensitivity, drop your LLOD, and allow those low concentrations to come into the linear range. And if you can do that, then you meet the criteria such as your coefficient of variance, your reproducibility, those types of things, your recovery, then you're able to get good accurate measurements below what you could do before, and now essentially fill in that missing data. So the Quanterix platform helps to do that. It definitely improves sensitivity in our hands, depending on the assay, anywhere from 4 to 700 to 1,000-fold. That change is really assay dependent. For cardiac troponin I, it had a large effect on our lower limit of quantification. It was around 700-fold. We can debate whether or not it's 700 or 500 or 1,000, but it's certainly a large increase compared to other plexes that we had been using. And what they have is a single molecule detection mechanism. Your antibodies are in very well-defined beads, which are very well-defined shape and size, which then will drop into these little very accurately wells that have been produced by Soma technology, and then essentially one antibody per one analyte, and you essentially count the beads that are fluorescent. It is a sandwich ELISA. 2 epitopes are being mapped for every analyte, which is an important distinction from some technologies that are only single capture and then do not have that second antibody to confirm or help to confirm the binding of the identification of your analyte. So single antibody or single apnea detection mechanism do not give you that second validation. Either you can do that by mass spectrometry, which I'll talk about at the end, or by having a sandwich ELISA. The Quanterix is a platform. You can mount different ELISAs up onto it, assays, as was mentioned earlier. So we took the cardiac troponin I kit. Quanterix makes this kit. We tested it, and the performance is really lovely. As you can see here below, we have standard curves from the kits. You can see this is 6 runs. They're on top of each other. You can see the low and high calibrator, one at 2.4 picograms per mil, the other at 10 picograms per mil. You can see just they come on top of each other time after time. It's really very well done. So we tested first in 20 healthy subjects. These are males, and we're just showing you over individuals, running them the same ones over 3 days. You can see they're coming in very close to one another. The CVs were coming in around 4%. We then extended that to 200 healthy individuals, and this is equal male and female. And what you're showing here along the x-axis is just the 200 individuals ordered in increasing concentration of the troponin I value that we obtained. And the healthy individuals were selected based on those values of including their B and P value had to be within the healthy individual. Below in red is the CVs that we obtained for each of these. These were all done in duplicate. If the CVs are outside of 20%, we normally redo them. But in this case, we just wanted to show you what we got on the first run. And what we were able to do is essentially obtain about 94% of healthy individuals were able to obtain accurate cardiac troponin I values, 94%. And you can see when you look at the lower limit of detection for ones like Abbott, Architect, their high sensitive TNI assay, which is in the clinic, and also Single-X, which is also a very sensitive assay, you can see that the Quanterix assay on the LOD is being able to obtain more information. It's the red at the bottom. Essentially, the LOD is 0.91 picograms per mil on this assay, and that's lovely. So we're able to capture more people. And if you go up to the limit of the 99th percentile, so the limit of quantification, you can see that it's the Quanterix assay that's able to pull in many, many more healthy individuals and get good reads on them, get accuracy both in recovery and in their CV. By using this assay. So certainly they've increased their sensitivity compared to other assays. It's very important because we're able to now get reads that are accurate on healthy individuals. Just in case you're making or wanting to make sure that we're doing those reads of the cardiac troponin I in the linear range of that sensitivity, that's what we show on the top or right, left-hand side. The red is the standard curve. You can see for all but one individual, we're able to be reading in that linear range. Interesting, in the pie diagram in blue, that's your lowest, the percentage of people with the lowest level of TNI under 0.1 picograms per mil. And then as you go from pink, green to purple, that's increasing concentrations in the healthy population. So again, we're getting a split kind of equally between people coming in at 0.1 out to 10 picograms per mil in the healthy, at least what we perceive to be healthy individuals. We validate this in the second cohort. I'm not going to show you that data. One of the interesting aspects of this particular assay was that we were looking at males and females. We had equal in amount. And just to remind everyone that our disease is different between males and females. Women have often MI is underdiagnosed, potentially for many reasons. But we wanted to know whether or not women just typically, even in controls, might have a lower level of cardiac TNI, so we're starting at a lower baseline, and that turned out to be the case. And this is work we did with Noel Berry-Mers here at Cedars-Sinai, looking really at the female to male. So again, these are healthy controls. males and females, 100 of each, and you can see statistically women have a lower level of cardiac troponin I at baseline even before without an MI. Whether or not that changes, whether or not this is a reason we're missing women, of course we don't know that yet. But interesting, there is also an age component. So what you're looking at here, we have 4 age groups, 20 to 30, 30 to 40, 40 to 60, and then above 70, male and female. So what you can see essentially is that, again, if you're under 40, you will differentiate as a female from males. And so males will increase over time, but women take a dramatic change upwards in their baseline level of cardiac troponin I as they age. So both gender and age are affecting the level of cardiac troponin I, and whether or not that, again, should be taken into account in the clinical domain, I think is something we should think about, but only if we have accurate and high sensitivity assays. Now you might say, well, what happens when you have an MI, which is what cardiac troponin I is really used for? And this is a schematic to show you that if you're coming in and your heart, that's kind of blue to red, it's ischemic. Troponin I may be leaking out, very low levels, maybe increasing with ischemia. And then, of course, as you advance into MI, you're going to have a rapid increase. And here's one example below that you would expect that if you have unstable angina or if you have non-cardiac chest pain, you might have these very low levels of cardiac troponin I picked up by Quanterix, that platform. But as you have MI, of course, those levels bump up and you're able to detect that with multiple technologies. So if we, this is work we've done with Fred Corley, a former wonderful student and postdoc, graduate student in my lab who went on to his emergency medicine at the University of Michigan. He provided us with some samples from his emergency ward for early MI, late MI, and no ACS, so no cardiac disease. And we were able to monitor, of course, the troponins very easily with the Quanterix, even those individuals with no ACS, because, of course, we can see controls. And why this matters is that really we do not know, well, we do know that there are release and clearance kinetics of people going into having an MI and then following an MI. And being able to follow them with accuracy will be very good. And what I've shown there with the dotted lines is really the 99th percentile for the Abbott and Single-X assay. So you can see that for the majority, this would be below it. Now, this is not to be misleading. Of course, 99th percentile is what everyone wants to be able to do, but that's not what we always use in the clinic. So we'll be able to pick this up. But the Quanterix, in this hands, at least we're able to get, we'll have no missing data. We can make better recommendations of what to do. So there are, again, other issues around the cardiac troponin I, and that's specificity and selectivity. And that's because TNI has post-translation modifications. And we've known this for a long time, that it can undergo modifications, and including proteolysis. And this means that the antibodies that are being used for the different ELISAs by the different companies may or may not pick up and pull down or detect all the different forms of cardiac troponin I. It may enhance it, it may make it worse. We don't know, because we don't know always the status of what the TNI looks like in the blood. And that's going to affect the performance of the actual ELISA itself. And this is probably true for most protein analytes. We just have not, as a field, looked into most of the forms that are circulating. But if we knew what the forms were, perhaps we could be smarter about the epitopes that we were making antibodies to. Having the technology to capture all of this maximally is going to be very important. And I actually think, obviously, because this is what our stick is in our lab, is that being able to exploit these proteoforms, these modified forms of these analytes might provide us more information about the person and give us a better understanding. But unfortunately, there's no shortcuts, and this is just a lot of work, and it needs to be done in my book. So the hypothesis is if you go, from example, into controlled tissues, you have a single analyte. As it goes to the disease progresses or gets more severe, you start to get these multiple forms. This is a degradation of a protein, and it very shows that you could imagine as these forms happen, you could understand the selectivity and specificity of where you are within that person's disease progression. This turns out to be cardiac troponin T, so often thought of an analyte that's not as susceptible to modifications as cardiac troponin I. That's not necessarily true. This is actually cardiac troponin T from a tissue of someone from 2 different regions of someone with having heart failure. So TNT also degrades in the plasma. Cardiac troponin seems to be a bit more stable, but these are analytes well measured. Of course, it's actually more complicated. This is actually showing you at the top from a biopsy all the different forms also of cardiac troponin I now equally degrading like T and troponin T did. At the bottom left, you can see a gel of showing all the forms in one individual's blood. This is from 2 microliters of blood, and you can see the cardiac troponin I is in various forms. And importantly, that the release and clearance kinetics seem to be different for these different forms. So, of course, what you're capturing in your ELISA may not be what you think it is and may affect the outcome of the actual value you obtain from your ELISA. More importantly, as I mentioned, could we actually use this diversity to increase our understanding of a specific person? But of course, both troponin I and troponin T are more complicated. They are actually a trimer in the tissue. They're TNI, TNT, and TNC. They can be released as monomer, dimers, or tetramers, and a lot of work has gone in to try to define that. There's certainly a TNI, TNC complex, but these proteins have very highly charged, certainly for cardiac troponin I. And troponin T has both a positive and a negative end to it, so it's also, even though neutral, has a tendency to want to bind to other proteins. So complicated analytes, modifications, degradation, I'll show you shortly, both phosphorylation and citrullination, as well as being able to be in different ternary structures. But we also know why this is important is that cardiac troponin I, even when you snip just a small part, even from the C-terminus, only 19 amino acids, if you remove it, you end up reducing cardiac contraction, at least in mice, by about 50%. And this was the work we did with Anne Murphy and Eduardo Marban when we were all at Hopkins together. And this tells you even small changes of the form of a protein can have large structural or functional effects. So being able to know which ones those are and to monitor it can add value to our understanding of a disease progression of a specific person. One could hypothesize that someone that has a full-length TNI circulating in the blood may be less prone to going on to have heart failure because probably it came from the heart. The heart's probably full of intact cardiac troponin I, even the ones that are still viable, the cell's still viable. Yet if you're seeing the degraded forms in the blood, if that's not coming from those serum proteases, which we know in this case it's not, if it's really coming and representing the form of the troponin I in the remaining viable myocardium as a degraded form, and a form that tells you or allows you to speculate that the heart is not able to contract as well. Perhaps those individuals are more likely to develop heart failure because the viable myocardium is under-functioning. But of course, we have not been able to prove that yet. So in this one, this is to look at the sequence of cardiac troponin I. All those arrows we showed several years ago, again with Anne Murphy and Pingbo Zhang, who was in our labs at that time, that there's more phosphorylations than anyone ever expected. The white ones were known phosphorylations at the time. The blue ones that we had determined just really a few years ago spread across throughout the whole molecule, and that's the structure at the bottom. The stars are showing you, and the squares and the triangles showing you where all those phosphorylation sites are found and across the whole molecule. Can they affect the epitopes? Yes. Of the antibodies being used in the clinical ELISAs, underlines are all the different epitopes which are, that are known for the different assays that are being used currently, and you can see a lot of them are in areas or near areas that have at least phosphorylation to some degree. But don't fret, because it turns out that many of these phosphorylation levels, at least in a subset of individuals, are not highly phosphorylated. They differentially phosphorylate, but not highly. So how can you go on to do protein quant? Well, there's mass spectrometry-based methods and ELISA-based methods, and now we have these ultra-high sensitivity ELISA platforms that allow us to push it even further. Mass spectrometry allows you to multiplex easily, allows you to specify very easily whether or not you want a diseased form or a total concentration. ELISA, that's a lot more challenging to do it, but we don't have the sensitivity in mass spectrometry, yes, even when we do capture followed by mass spectrometry. So ELISAs are not going away. But let's talk a little bit about mass spectrometry. What we do is often, not always, but often in this case, we proteolyze the protein, TNI in this case. We're going to just target a specific peptide. The selection of peptides actually are very complicated, takes a lot of work, but it's like making an antibody. Once you have a tool, it's a tool and you can use it easily. So in this case, we're monitoring one peptide to monitor cardiac troponin I. When we do discovery, something like data-independent acquisition, we're measuring all peptides in our sample, so thousands of proteins, thousands of peptides, and we're monitoring that one peptide for cardiac TNI. When we do something called targeted multiple reaction monitoring or selective reaction monitoring, they're the same thing essentially. We essentially exclude all other peptides in our mass spectrometer and trap only the one of interest. So we get rid of everybody else and trap the one peptide for TNI. And then we quantify it by using an equivalent heavy labeled peptide. You spike in X amount of the heavy standard, and then you measure the amount endogenous, and you're able to get a quantitation in the same way you do with an ELISA. Very powerful method, but we can do it both in DIA and in by targeted assays. So we did that, and we went in after all the phosphorylation sites, lots of little peptides. This was challenging for cardiac troponin I, plus a peptide that was unique for total, so it was not modified. That's that red one that's labeled. And then we went in into essentially made a multiplex, so each peptide we targeted, we eliminate all other peptides except the one we want as they're being put into the mass spectrometer one at a time. So over time, you end up getting different peptides coming out, and you essentially can quantify them compared to a standard that you've spiked in. Just like an ELISA, you need to make sure you're in the linear range of the concentration. You need to check your CV and your percent recovery. That part is no different. That accuracy has to be there just as it is with the good ELISA. So we did that, and essentially this take-home message here is that we were looking at controlled tissue and two different types of heart failure, is that we were able to quantify both total as well as each phosphorylation site. The take-home message is that really there's a lot of phosphorylation, but it's at very low level. But there are sites that did change whether or not you had heart failure. So again, you need to know which sites to go after in order to make this useful, but you have to be analyzing what's present, and whether or not these are going to affect your epitope will depend on the individual and the extent of phosphorylation. But, of course, that would be too easy. TNI, again, just like many other proteins I'm sure we just don't know about, it can have other modifications. It's known to be acetylated, but one I thought you might be interested is that citrullination. This is a deamidation of arginine residues by an enzyme family called PAD, peptyl arginine deamidases. And essentially it goes in and removes a nitrogen group from a side chain, making the amino acid going from an arginine with a positive charge to citrulline in the protein itself, which is neutral. So it goes from charged to neutral, essentially neutralizes arginine. This is a modification well known in autoimmune diseases. In RA, citrulinated peptides, citrulinated forms have been thought to be involved in the autoantibody response of RA. And interestingly, people with RA, rheumatoid arthritis, are at higher prevalence of getting a heart attack. This is work we've done with Joan Bethon out of Columbia University, and she was the one that came up with this great hypothesis. And the hypothesis was, well, maybe in the heart citrullination happens, and if that was true, maybe some of those citrulinated proteins do leak out over time. And now we can think that it's very possible, since we know that there is leakage of proteins like cardiac TNI even in healthy individuals. And maybe these modified forms, if exposed enough times, would produce an autoantibody response in heart failure. Well, worked with her and with Justina Furt-Bauer in our lab, this turns out to be a pretty good hypothesis. And we worked on the mass spectrometry in order to find a good way to find citrulinated proteins. It's not an easy PTM for mass spectrometry, but we did it, and we looked at patients who have heart failure versus control and found proteins which are citrulinated. It's not a modification like phosphorylation that is, happens to many, many proteins, like a signaling cascade. Citrullination is very rare and happens very specifically, and that's what we know based on the data. But interesting, when we did this, what's shown here is a map of all the proteins, or a subset of the proteins we found to be citrulinated differentially between control and heart failure tissue from individuals, from patients. And you can see that there are different proteomes. So there's mitochondrial proteins which are citrulinated, but there's also myofilament proteins citrulinated, and yes, cardiac troponin T and cardiac troponin I are citrulinated. Interesting, there are not a lot of citrulinated proteins on those. This is showing you the proteins in the myofilaments, which are citrulinated in either common, which are, or those which change with the two different forms of heart failure. But at the top, you can see in the box that troponin I has two arginines that can be citrulinated. Now, this is quite remarkable because cardiac troponin I has a PI of 9.5 and is very, very heavily enriched in arginines, but only two of them are actually citrulinated. And troponin T is also citrulinated. So this can happen, happens in heart. And you can imagine a subset of this might be actually released into the blood. So the question then becomes, could you actually be producing autoantibodies from the citrulinated in a subgroup of individuals? And that was the question we wanted to know. So we did a second method, and this was with Tim Barter out of his company, and Joan, ourselves, and Justina, where we took tissue from hearts, from failing hearts, from control hearts, fractionated them in, well, first of all, we took those, and then we induced citrullination maximally by adding the enzyme in the presence of calcium. So we had two samples for both controls and heart failure pools. One of them was just naive, and the other one maximally citrulinated. We then took those pools and fractionated them into about 1,000 fractions, arrayed them on an array, just as most people do, and then passed patient serum, patients who have RA with differing extent of cardiovascular disease. Then we took those which kept lighting up and went into those fractions and asked, are any proteins citrulinated? If they're citrulinated, maybe that's what's causing the autoimmunity. To validate that, we would take those proteins that we thought were candidates that were citrulinated in the autoimmune reactive fractions, and then spot those and see whether or not in their citrulinated form and naive form, and find out whether or not they were lighting up. So we did that, and several proteins were identified to be citrulinated, to be autoimmunogenic in the serum of individuals with RA. And that, of course, turned out to be cardiac troponin I, cardiac troponin T, and myosin heavy chain, among many other proteins, and it was the citrulinated form. So next what we did is we took those and spotted modified troponin and unmodified troponin. So normal troponin, and then troponin that was treated with PAD to induce citrullination. We took myosin that was naive and myosin that we induced maximum citrulinated, spotted those, put a patient sera against it. And what you can see here is that the signal really came from the citrulinated form. So we just did troponin, so we don't know if it's troponin I or troponin T, but what this infers is that we can have troponin I, citrulinated forms, or antibodies in RA individuals who are reactive to citrulinated troponin, and they're producing autoantibodies. So what could this all mean? Well, we can suspect, and this is an idea that actually came from our work with ImmuneArray that works on traumatic brain injury, and this was with Alan Everett out of Hopkins, and the concept of being able to measure the antigen upon the injury, for example, cardiac troponin I, and troponin T as a reflection of the injury. But if you add in the post-translation modifications, perhaps you can get an indication of what will happen in the future. You could also monitor if those proteins are citrulinated, and if they are, they could be autoantigenic. And if they're autoantigenic, then you can also monitor and capture the autoantibodies as a late-term effector along the disease. So this is a new concept. Doesn't seem like such a new concept. We've been talking about it for a few years, but it is a conceptual idea that whether or not we could use these high sensitivity assays to be able to monitor the protein, the modified protein, the potential autoantigen, and the autoantibody, and you'd want the best sensitivity possible because you'd want to be detecting the smallest amount of these as an indication early on, and then watch it increase over the lifetime of the individual as a prognostic indicator. We'll see if we're able to do that. So, last slide. So is one protein enough? So I've talked about TNI in all its forms, and TNT in all its forms, phosphorylated, degraded, citrulinated, potential for autoantigens, being able to use those as a marker of injury inside the heart, prediction and production of autoantibodies. But that's one protein. Can you do, and do we need panels? We often talk about panels. I think we do. I mean, well, I'm sure we do, especially in chronic diseases where it's more than just heart failure. You have diabetes, you have the natural history and aging of an individual, you have early atherosclerosis, and this is where we've made panels, for example, this is a 73 protein mass spectrometry multiplex. So we run and quantitate 73 proteins. One of them is a control, so 72 plasma proteins, and they represent essentially major cardiovascular disease indicators. So they indicate atherosclerosis, lung injury. It indicates immune, inflammation, atherosclerosis, kidney function. By monitoring all of these 72 proteins and a given time in essentially 5 to 10 microliters of blood samples, we're able to assess an individual overall the time. Is this cardiac troponin I? No, but this is a step that allows us to very easily monitor people to find out whether or not they're at risk and then shuffle them into something like the ultra-high sensitive TNI assay. So this is a new plex that we've made over the time. It was done by Chin-Fu in my lab, and we're now using it both for venous and collection as well as for remote monitoring devices, as well as with an ability to measure overall health of an individual. So where are we? What's our summary? Well, our lab proposes, as do many others, that accurate quantification of proteins, whether as a single protein or as a plex, or as a single protein and its modified form or its autoantibodies, is going to allow us the specificity that's needed to really individualize who you are in the progression of a disease and across the multiple disease pathologies that you might have going on in your body at any one time. That if that is true and that's going to be enablement of precision medicine, then we have to step back and say what is truly needed. Well, what's truly needed is that we need the sensitivity of our technical platforms. We need to have the selectivity of our assays, whether or not that is making sure that we don't have interferences and that truly we're measuring a total concentration or exploiting the modified forms of a protein, and the selectivity of the disease setting, whether or not we only want to be measuring MI, or do we want to be measuring all of their wellness, whether as you lead up to an MI, what their atherosclerotic status is, what the inflammatory status is. We need to make sure we have the selection that's appropriate for the disease spectrum over the lifetime of an individual, both combining these broad panels that look at multiple pathways, as I indicated from the 73 protein plex, versus something like cardiac troponin I, versus something like the troponin in all its modified forms. So I think we need to be able to put this together with both the accuracy of the measurement, making sure we really are measuring the analyte that we say we're measuring, the ability to get the sensitivity where we need it, using things like the Quanterix, being able to multiplex out as we can do more and more with mass spectrometry, but of course not with quite the same sensitivity as we get with the ELISA platform. And somehow put this into a business model that allows us to actually reach out past academics and past the clinical chemistry labs into a form that allows us to take this to the next generation. So of course this is done by not just me, but an amazing group of collaborators and an amazing, amazing laboratory. Those individuals in red are ones that have really been involved in many different aspects of this thought process and will continue to do so. I'm also very grateful I get to work with amazing scientists in a large number of companies to try to push this vision forward. And in our next step, as we try to put together additional labs that help us translate discovery all the way through into clinical and remote monitoring devices, I think I'm very excited, and I think the future is going to be only better. So thank you, and I'm open to any questions that you might have. And again, I appreciate you spending this hour with us.
Okay, thank you very much, Dr. Van Eyck, for that excellent talk and overview. There are a number of questions from the panelists, and we have a few minutes left, so I'm just kind of ask you the questions as they've come in during the webinar. So the first question is, do you see value in kinetic biomarkers, i.e. quantifying the production of or clearance rates of target proteins and molecules rather than just static biomarker quantitation?
Oh, absolutely. What a wonderful question. This is a great question. Someone's thinking very hard and very, very wisely. Absolutely. I think this concept of moving from static to this dynamics is very important, especially on the acute stage. And we just are trying to get published a paper where we do this for brain natriuretic peptide, so BNP, which of course is a great marker for heart failure. It's released as a prohormone, which then gets cleaved into the really functional tiny peptide, the 32 residues long, which is biologically active. The moment it hits plasma, it starts to degrade. And understanding how fast that degrades in different individuals actually can tell you a lot about what is the status of the peptide that actually binds to the receptor, which is the biological readout. So when this 1 to 32 peptide binds to the receptor, its specific receptor, that's when you get the natriuretic effect. So it's very important to know the concentration of that protein. But of course, as it gets degraded, its affinity for the receptor drops, and at some point, that peptide, even if you're measuring it in your ELISA, is not biologically active. So I think that things like that are just so good for being able to understand the mechanism of the biomarkers. And if you believe, as I do, that you want to get as close to the biological activity for your biomarkers, then this is a type, understanding this kinetics is very important. The other part of your question, which is release and clearance kinetics, I think we need to understand those very well. And that's why in our 72 protein plex, I'm very happy that we have 2 kidney markers, because I think those kind of clearance, not to say that markers just get cleared through the kidney. We know that's not true. But understanding those dynamics and how they're influenced by gender, ethnicity, age, I think is all part of this understanding of molecular signature. I do not understand how you can have precision medicine and talk about having precise measurements of individuals or signatures of an individual without understanding the clearance and release kinetics of every biomarker and the dynamics of them over the short term as well as over their length of the lifetime. So wonderful question. Thank you for asking it.
Okay, next up, there's actually 2 questions that are somewhat related, so I'll put them together, I think. It comes from one of the panelists or the guests. Can routine monitoring baseline cardiac troponin I levels be used to predict risk for MI? The first part of the question. And then what is the current state of acceptance of high sensitivity troponin testing for heart health monitoring?
Another great question. So we do not know yet whether or not monitoring these baseline low levels in cardiac TNI in patients over time, whether or not that will provide you with a risk prediction. I think it's a good hypothesis. I think it's a strong hypothesis. What I can tell you with Noelle Barry-Merz, we have not published this yet, she had samples from the WISE consortium where we had age matched, these are women, so genders already matched, at baseline, and then within 10 years of those individuals who had a CVD event or did not. And we measured those individuals that had the CVD event, so this is measuring 10 years ahead of time, their levels were higher, slightly higher. The problem is that they still overlapped with normal levels, so it's not a pure distribution, but they were significantly different. So this is an indication that we could. I am of the belief, as many groups, including people like Lee Anderson and SISCAPA, that if we can do this continuous monitoring of individuals, let's say once a year, once every 6 months, we don't know, but certainly of mid-risk individuals, that we'd be able to risk predict. And in fact, California Precision to Accelerated Precision grant we just received with, again, Noelle Barry-Merz and Brennan Spiegel, we're trying to do that. We're trying to enroll mid-risk individuals for CVD and then to monitor them over a period of time, both with this Quanterix TNI, BNP, and then the 73 protein plex that I mentioned to you at the end of the talk. So I think these are early days. I think it's a great hypothesis, and I'm hopeful, which is why I have such a hypothesis. So nice question. Thank you for asking.
Okay. Next question is, how much of an issue is sensitivity for immuno-MRM versus ELISA? Is the dynamic range the bigger issue for MRM? It says mass spectrometers per se have shown to go beyond zeptomoles on clean samples.
Yeah, so this is a great, so with immuno-MRM, you can do it 2 ways. You can immunoprecipitate the protein and then, for example, do it plus the modified forms. That's what we're trying to do with TNI, to pull down the TNI and then to have assays to the different forms. I can tell you it's tricky at that level. TNI is we can get it for MI. If you have an MI, we can see it. Not a problem. It's to get below MI that's really challenging. It's just, I'll talk about this selectivity. The second way, which a lot of people are having success, is to go after the peptide, make antipeptide antibodies. And again, it's going to come down to, just like ELISA, how good is your antibody? The mass spectrometry has a dynamic range. So if you enrich a protein or a peptide into that linear range of the mass spectrometer, your assay, just like an ELISA, will be accurate and precise. The real trick is really the capture of the peptide or the protein in a complex mixture. So you can imagine how good your antibodies have to be. They have to be as good as an ELISA, because you need to, we all know that an antibody or any protein will bind to anything at some binding constant. And in blood, we have such a large dynamic range from albumin down to something like TNI. So if we want an antibody to pick up TNI, and it's at, picograms per mil, and albumin's there at, 80 mgs per mil, it's very hard for an antibody not to also bind to some affinity to some peptide in things like albumin. And this is where the challenge comes, and why sandwich ELISAs and immune MRM assays work well, because you're using a second device or second method to determine that you actually have bound the analyte and you've actually quantifying off that detector. So in an ELISA, it's a secondary antibody, second antibody. MRM, it's your peptides that you're now targeting in your mass spectrometer. So I think, again, you'll see some immune antibodies come out, but in a clean sample means you don't have albumin and a billion other proteins hanging around competing for your antibody to make it worse. And that's the challenge we're having with TNI, for example. We have great commercial antibodies used in ELISAs, but getting it to pull out of, several hundred microliters of blood, which of course is what's required, is challenging. So I think this is technology that's going to come up and really push the limit of what we're going to be able to do. And, people are very smart and innovative, and they're just going to get these things to be better and better. And the real question comes in, where would an immuno-MRM or an MRM assay be useful, more so than the current ELISA platforms that we have in clinical labs? And there are several indications, things like post-translation modifications, which are easier to do if you have mass spectrometry. Could be, genotyping, plexing, or it could be in things like remote monitoring, where proteolysis of the proteins help get it out of the devices. So lots of interesting things over the next few years. Great question. So is there one last question we can have before we tune off?
Yeah, I think there are a couple more questions. So let's ask one more question, then we'll finish up. The next question I have is, are there any other inflammation-specific biomarkers that are important for the detection of cardiac disease?
Oh, absolutely. I think that's been shown that often CRP, of course, is the classical one. It's been shown that if you have CRP, TNI, and BNP, you're at higher risk. So if all three of them are elevated, you're like in the worst circumstance, has been shown and known for a long time. And the problem with CRP, of course, is it's up as it should be in many, many diseases because it's your general inflammatory response. But there are many other proteins, including high abundant proteins, things like FAHA and a whole bunch of other ones, which I can't recall off the top of my head. But there's many inflammatory markers or pathways that can be seen in high abundant proteins. But also, of course, there's the cytokines and chemokines that are all involved. So, understanding how that changes in an individual and which of the specific pathways within inflammatory responses worse is not clear, but we do know that if CRP is elevated and you have TNI elevations, even below MI levels, so at these baseline levels, although not as sensitive as Quanterix is with higher levels, but not an MI diagnosed level, that we know you're at even higher risk. So your body tells you these things. It's just we have to understand and learn to listen to it, both with the biomarkers that are circulating, but of course also with imaging and other modalities that we are using to understand the heart and the whole vascular system. Thank you for asking that one. That's also a very great question.
Excellent, Dr. Van Eyck. Thank you very much. And on behalf of Quanterix, I'd like to end with a comment that somebody made on the panel, which was specifically wanting to thank you very much for a very enlightening and interesting lecture. So thanks very much for your help, and I guess I'll turn it back over to Elizabeth.
My pleasure.
Thank you very much, Dr. Van Eyck, for your presentation. thanks to Kevin Hrusovsky and Mark Roski for moderating our questions. And thanks to all of you for coming and spending some time with us. I hope you found some answers to your research questions. We appreciate you coming and would like to wish you all a good day.